Hello, biology students. Today we're going to be using our DNA knowledge to talk about how DNA replicates or makes copies of itself. But before we do that, we got to learn a little bit more about the DNA history. So, to learn about the DNA history, we have two pairs of people that we're going to be talking about really quick, and we'll learn about these people again in class. So the first pairing is Rosalind Franklin, that's a female, and Maurice Wilkins. These two people were the first to study the double helix shape. Before we knew that DNA was in the shape of double-stranded twisted ladder, people thought it might be a single strand. Some people thought a triple strand. Well, Rosalind Franklin, even though back then in the early 1900s, she was a woman and it was really hard for women in science to get a lot of recognition, she was able to do a technique called X-ray diffraction and make this image using an X-ray. And in the image we see two crosses or an X-looking shape, which you should draw because this shape sometimes actually comes up on the SOL. So this shape suggested that instead of a triple helix or a single helix, because there are two X's, that it was a double helix. But because Rosalind Franklin was a woman and Maurice Wilkins wasn't very famous, two young scientists, James Watson and Francis Crick, very well known as Watson and Crick, right? They're very famous. They end up stealing that picture, and it's the last piece of their puzzle to make this three-dimensional diagram, which helped them realize that the double helix, in fact, was the shape. They were faster to publish their article in a paper, and they are the ones that end up getting credit. So poor Rosalind Franklin doesn't get any credit. Nowadays, we know she helped, but back in the day, she did not win the Nobel Prize like these two did for figuring out that DNA was a double helix. But we're talking about DNA as a double helix because that shape, the double helix, is what makes DNA copying or DNA replication very doable. When we say we're replicating the DNA, it means we're going from one double helix and we're going to use that single double helix to make two identical double helices. This is the plural version of helix. Okay, how can we use one to make two? Well, really, this is going to happen before mitosis. Before a cell can divide, we had to copy the DNA. And we already know what part of the cell cycle this happened in. It happens in interphase, right? And it has to happen so that the DNA in both of the daughter cells after mitosis is identical. Well, we do this by using DNA as a template for making more DNA. What do I mean by template? A template is a pattern to make more copies. All right, back in the day before we had cool digital photography, there would be these things that kind of were funny colored that were called negatives that would be made by regular cameras. Those negatives or film strips, we would be able to send those to a photography store and they would make copies over and over again of more photographs. Right? That's what a template is. It can make many copies. Well, DNA, one strand can make two DNA copies. So we call DNA a template because one part of it can make two copies. That's why we call it a template. Well, how does this process really happen? That's the details of what we're talking about today. We're going to practice together in class. So there's going to be 3 steps to this process, and you're going to want to get down the major players and the major steps. All right, we're going to not need to make lots and lots of detailed pictures because we're going to have some pictures given to us in class, but you might want some basic pictures. The first step is to untwist and unwind the DNA. Untwist and unzip is another way of saying it. So the DNA double helix, remember, is that twisted ladder shape. So we got to unravel it, untwist it, okay? And there's an enzyme that does that called helicase. I remember that the helicase enzymes always end in this phrase, ace or in. It's helicase because it undoes the helixy looking shape. And it does that by breaking the bond. So it'll untwist it. First, make it into more of a ladder-looking shape, and then it'll actually unzip it. Think about a zipper really being pulled up, right? Breaking those hydrogen bonds between the different nitrogen bases. We're going to practice seeing this in class again, and you guys are even going to try to do it yourself. The second step is that this is all happening within the nucleus. Well, the nucleus is not full of just DNA looking like this. It also has the DNA building block, the DNA monomer. Okay, we know the monomer of DNA is called a nucleotide. So there are free nucleotides sitting in the nucleus, and they will slowly be added to the DNA strand. Again, here we have a DNA template. So this G, what will it probably want to match with using our base pairing rules? It'll want to match with a C. And those matching bases, they're just floating around in the soup of the nucleus, are just going to find their matching pair. So we call those floating around nucleotides free nucleotides, and they'll be added to the open strand. So we call this the open strand or the template, because that template is going to help us make a copy, adding the correct bases using base pairing rules. All right, but because this needs to happen fast, what kind of thing helps us have chemical reactions happen very quickly? You're right, an enzyme. And the enzyme this time is called DNA polymerase because the DNA is forming a polymer, many monomers. So we add many monomers called a polymer, and we use base pairing rules to properly match them up. So those nucleotides just floating around in the soup of the nucleus will add to the open template strand. Wonderful job. We will practice this in class. And at the end, we've done that to actually the both sides, since there was an open side over here that was unwound and there's an open side over here. Notice that we have the gray representing the old strands, and here we have the red representing the new. The new stuff, right, will form on each of the two open strands, and at the end we'll have two identical double helixes. Okay, so the end results, we check that they're really identical. We have a phrase for this fact that there's some of the new stuff, some of the new double helix is the original, and here it's pictured as blue, and some of it is pictured as yellow, the new pieces. We call that semi-conservative, and the definition is really important, so make sure you write it down. Each new double DNA molecule or double helix has one half or one new strand, which here it's yellow, and one old or parent strand, and here that's blue. Let's double check that it's that way on both sides. Why do we have a name for that? Well, because scientists didn't always agree that it would be like that. Sometimes they thought it would be all chopped off, part new, part old on both sides. But really, it's really clean where the old template matches with a new strand. Wonderful job, guys.